So in this second part, we talk about the application of the EEG and brainwave research to understand the functional role of oscillation in perceptual and cognitive processes. So as you know, perception is our ability to capture the information that our senses receive, and cognition is our ability to interpret them. So we'll start, okay, our second part of the talks with Maëlan Menétrey, that we talk about the alpha oscillation in visual perception beyond rhythmic sampling. Then we'll go to the states, and we will have Mattia Pagnotta that will talk about the working memory, the relationship between working memory and alpha oscillation. And after him, Martin Seeber, we talk about also about theta dynamics of real world and imaging navigation. So we will have some information also about another brain rhythms that is not alpha. Okay, so here. Okay, so here we have our three speakers of this afternoon. So we will start with Maëlan Menétrey, that was a bachelor degree in psychology from the University of Lausanne, and 2 master degrees in neuroscience and psychology from the University of Geneva. Between 2020 and 2024, he completed a PhD in neuroscience under the supervision of Professor Michael Herzog at the PFL. Recently, he began his postdoctoral position with Professor David Pascucci at CHUV and UNIL. Using EEG recordings, Maëlan investigates brain dynamics both at rest and during perceptual and cognitive tasks, aiming to identify specific neural features that predict behavioral performance. The stage is yours.
Thank you for the introduction. Hello, everyone. And so today I will present, so now I am a postdoc in the Psychophysics and Neural Dynamics lab, but I will present some work that I did during my PhD at EPFL. So as we saw already in the previous talk, the brain is a rhythmic organ, and this has been observed since 100 years ago when Hans Berger invented the EEG, the electroencephalogram, and measured for the first time some brain electrical activity. And in particular, he observed some prominent waves around 10 Hertz, which he called the alpha oscillations. Since then, alpha oscillation has been investigated a lot, and we know now that this oscillation reflects some neural synchronization and pulsed inhibition. First, when you measure a strong alpha power, it means that neurons are periodically activated and silenced. And this also implies that the phase of the alpha oscillation will reflect one excitability, one phase with high excitability and one phase with high inhibition. And we use also a lot the frequency of the alpha oscillation as a measure to determine the duration of each cycle and the distance between two highly excitable phases. So the brain is rhythmic, but perception is also rhythmic. For example, with the flickering wheel illusion, we can have an idea of this phenomenon. If you focus on this fixation point, if you look at this fixation point, you might have the feeling to perceive some flicker in the center of the wheel. I hope it works in this room. and this experience could be the result of fluctuation in perceptions. And some studies have shown that the illusory flicker that participant could perceive was stronger when participant exhibited strong alpha power, and also an individual with a fast alpha frequency would report a faster illusory flicker. However, the links between alpha oscillation and rhythmic perception remain relatively unclear. First, there are at least two main potential effects. Alpha oscillation could act as a sensory modulator. For example, we know that when we present a brief flash, successively, so over several trials, sometimes the flash will be perceived and sometimes the flash will be missed. And it has been suggested that alpha oscillation could partly explain this variation in the perception. First, when alpha power is strong, we have therefore this favorable phase, highly excitable phase, and a phase associated to inhibition. And this could explain that when sometimes the flash is missed because it coincided with the highly inhibitory phase. In contrast, when the alpha power is low, this reflects high excitability, and therefore, regardless where the flash is presented along the phase, it's more likely to be perceived. But there is also a theory that goes even further, suggesting that alpha oscillation could integrate information. For example, if you present two successive flashes with the same interval, several times one after the other, sometimes these two flashes will be perceived as a single one. And it has been suggested that this is directly related to the alpha oscillation. If the two flashes fall within one cycle, then they will be integrated, only one single flash will be perceived. But if the two flash falls in two different alpha cycles, then they are more likely to be segregated. So these are some suggestions linking brain oscillation to perception, but it is also still unclear what aspects of perception are modulated by alpha oscillation. First, it is possible that it's only the content of perception, what is perceived, that is affected by alpha oscillation. And one explanation could be that it is the unconscious processing that is modulated by alpha oscillation and affect the content. The other hypothesis is that it's not only what is perceived that is affected by alpha oscillation, but also when the percept occurs. This alpha cycle will define some moment where there are conscious percept, and it will really predict the discrete percept. What I will present today are two studies. First, we tried to investigate whether alpha oscillation modulates conscious perception or unconscious sensory processing. And then we wanted to test this idea that alpha oscillation determined a window of temporal integration. First, let's start with the first study, in which we use some pre-stimulus alpha activity analysis. Pre-stimulus activity is the activity, EEG activity, that you will measure before the onset of a stimulus. And we know that, as I said before, when you present some stimuli, sometimes it is some brief, very brief stimuli, sometimes it is perceived, sometimes it is missed. And for example, in one of a classical study showing this, they were presenting a brief flash, either on the left or on the right of a fixation point. And then they look at the brain activity in trials where the flash was perceived and in trials where the flash was missed. What you see here is a classical time frequency representation of the brain activity. The black line corresponds to the onset of this flash. And it is known that when you compare brain activity in between trial where the flash was perceived versus trial where the flash was missed, you can find some difference in brain activity, significant difference in brain activity before the presentation of the flash. And in particular, in this study, they showed that they found a difference in alpha power before the presentation of the flash. When the flashes were perceived, alpha power tend to be lower in the pre-stimulus activity. And it is interesting to note that this effect is observed several hundred milliseconds before the onset of the flash. However, one limitation of these studies is that they use very brief stimuli and static stimuli, and we don't really understand how long lasts the effect of alpha oscillation. Also, these studies usually ignore the dynamic nature of perception and the fact that information needs to be integrated over time. So in this first study, we ask whether a pre-stimulus effect could also be found in tasks where visual information is integrated over longer time window, and in particular longer than the duration of one alpha cycle, so more or less 100 milliseconds. To do so, we used the sequential meta-contrast paradigm, which produces some long-lasting feature integration. The idea is quite simple. You present a sequence of lines starting from the center to the screen to the periphery, which produce the percept of one moving line. We know that if you add this vernier offset, so the one line with a small shift, this offset will be integrated to the following straight line, and the participant will perceive the offset moving from the center to the periphery. And when you add later in the stream an opposite vernier offset, in this condition, the information will be integrated and participant will perceive only one single offset moving from the center to the periphery. This suggests that first, the brain unconsciously processed all the individual features, but what we consciously perceive is only the integrated output. So using this vernier anti-vernier condition with two opposite offsets, we recorded EEG activity, and we asked participants to report the perceived offset in this vernier anti-vernier condition. So sometimes the integrated output was a vernier. For example, here, let's say that it's an offset, a left offset, because the lower bar is on the left, which corresponds to the first central vernier. And in other cases, it was the second vernier that was dominating. So participants always have to respond, to give a response corresponding to one of the two offsets. And what we observed in the pre-stimulus alpha power was that it was still possible to predict whether the first presented vernier or the second presented vernier dominated the integrated percept. In particular, we found that when pre-stimulus alpha power was stronger, the first presented vernier was more likely to dominate the integrated percept. And this effect was found mostly in the back of the head. And again, what you can see here is that the effect, the significant difference in alpha power was observed several hundred milliseconds before the onset of the SQM, here between 450 milliseconds to 250 milliseconds before the onset of the SQM. So this kind of effects are quite puzzling, because since alpha cycles are only more or less 100 milliseconds, it is weird that several cycles happen between the effect in power and the actual presentation of the SQM. So this leads us to an additional analysis where we try to investigate the following changes in alpha activity just after the significant difference in power that we found. So the significant difference in power that we found is highlighted with this box here. And what we observed was that following increase in pre-stimulus alpha power, there was a decrease, a slowing down of the frequency of the alpha frequency. And this particular slowing down of the alpha frequency coincides actually with the onset of the SQM. And we also show with the mediation analysis that the alpha frequency mediated the effect between pre-stimulus alpha power and the reported offset. So this first study suggests that alpha oscillation really modulates unconscious processing, because in the SQM, what is perceived is always what has been integrated. And since it's impossible to perceive the two offsets separately, then this suggests that the pre-stimulus alpha power and the frequency affected the integration of the stimuli. And also, this study shows that following the increase in alpha power, there is the start of a cascade of neural changes, and here maybe at least a slowing of the alpha frequency. And we can therefore summarize this result with this little sketch. Following higher alpha power, there is a slowing down of the frequency during the next cycle. And maybe because the alpha frequency is slower during the SQM presentation, this modulates and make the first presented Vernier more represented in the integrated percept. So this was the first study, and then because also the SQM last over here in this condition, the SQM was more or less 200 milliseconds. It also suggests that information was integrated over longer duration than the single alpha, just one single alpha cycle. So this is what we wanted to test again with another study to really test whether alpha oscillation determine temporal window of integration. And here we use the individual alpha peak frequency as a measure. The individual alpha peak frequency is the strongest frequency, the frequency with the strongest power within the alpha band that we can record in individuals. Here we use resting state individual alpha peak frequency. And we know that different individuals, when you look at the power spectrum, can have different peak in the alpha band. And we also use this individual alpha peak frequency because several studies show that, again, with this example of two brief flashes that are presented successively, the interval between the two flashes that are needed to be able to segregate the two stimuli can be predicted by the individual alpha peak frequency. So usually when you have a fast alpha peak frequency, you have a better temporal resolution, and at least this is how it is presented, and you are able to segregate two flashes even when the interval of time between the two is short. However, again, one main issue of most of the studies investigating and linking individual alpha peak frequency to temporal resolution of perception is that they use usually two stimuli that are separated by an interval of time which is less than 100 milliseconds, so less than the duration, at least never exceeds the duration of one single alpha cycle. And again, we were wondering whether this effect of individual alpha peak frequency on perception could still be found in tasks where two stimuli are integrated, even though the interval is longer than 100 milliseconds. So again, we use this task that you already saw several times today, the visual backward masking. We had these 4 conditions. In all the conditions were randomized, and participants always had to report the offset, the direction of the Vernier offset. And so in one condition, the Vernier was presented alone. In one condition, only a mask was presented. So in that particular condition, participant had to guess. And in 2 conditions, we had a Vernier and then a mask. The mask was either presented right after the Vernier or presented after 120 milliseconds. What is important to understand with this task is that the ability to report the Vernier offset in the 2 conditions with masking depends on the ability to segregate the target from the mask. And so we hypothesize that if individual alpha peak frequency can affect this ability to segregate the Vernier from the mask, the effect should be observed only in the condition where the Vernier is directly followed by the mask, because it's the only condition that in which the width of the alpha cycle could affect whether the Vernier and the mask fall within the same cycle or not. In the long SOA condition, the Vernier and the mask are always presented in 2 different alpha cycles. And to be sure to have individual with low alpha peak frequency and high alpha peak frequency, we had the chance here to have data of healthy control, but also of schizophrenic patients. And we knew from the literature that schizophrenic patients typically exhibit slower alpha peak frequency. And this is what we reproduced with this data. There was this significant difference in the individual alpha peak frequency. So now the main analysis of this study was to try to predict visual backward masking performance using the individual alpha peak frequency and also adding the group effect and the condition effect. And what we found, it was that we had, of course, this significant difference between healthy control, the blue line, and schizophrenic patient in the three conditions. So schizophrenic patient always perform less well than healthy control. And we had also a significant effect of group, of condition, sorry. So in the two conditions with the mask, the performance is lower than in the condition with the vernier only. This is not so surprising, but we found that alpha peak frequency also predicted significantly the performance. And because the effect wasn't found with any interaction with the group or with the condition, this suggests that the alpha peak frequency affected similarly the performance in the three conditions, so also in the vernier-only condition and also in the long SOA condition. And also, because we don't find interaction between the individual alpha peak frequency and the group, this also suggests that the effect is similar between in healthy control and in schizophrenic patient. So if we link that to what Dario presented before, there is a difference in the individual alpha peak frequency between the two groups, but it's not this difference that affects performance. Here the performance is modulated by the speed of the alpha oscillation, regardless whether the participants are schizophrenic patient or healthy control. So it's a general effect, and it's why we suggested that individual alpha peak frequency modulates general visual performance instead of specifically temporal resolution and processing that are constrained within the duration of one single alpha cycle. And one of our hypotheses was that alpha frequency rather affects the processing duration. Here again, a small sketch to summarize the finding. Even when the target, the vernier, is presented, the target is preceded by, no, even when the mask is presented after more than 100 milliseconds, the speed of the alpha still matters, and it is therefore possible that individuals with fast alpha rhythm are able to process faster the target stimuli, which is therefore shielded from the disturbing effect of the mask. In contrast, when the individual exhibits slower alpha rhythm, because the processing duration is longer, there is more the different sensory, the successive sensory signals are more likely to interact. So the key findings of the two papers that I presented are first, this idea that alpha oscillation modulates unconscious processing over longer time period than what is usually suggested, and also that the alpha frequency does not dictate a specific window of temporal integration. And together, I suggest that these findings challenge the idea that alpha oscillation really define discrete frame in the structure of perception. Instead, they just modulate the unconscious sensory processing, which ultimately affect the content of perception. And one implication of this result is that the frequency, the frequency of alpha oscillation also modulates the excitability of the neurons. For example, we know that alpha power will modulate the duration of what is called the duty cycle, this phase which is associated with high excitability. And similarly, we propose that alpha frequency could have the same effect. So an individual with slower alpha frequency will also have larger duty cycles. And in this context, in the context of temporal processing, this could increase the neuron responsiveness. An individual with slow alpha frequency will respond, the neural response will be stronger, and this might increase and prolong the persistence of sensory traces, mixing the different informations. And therefore, the alpha frequency could be an index of the efficiency of visual processing. And in these two studies, we show that we can at least measure this in two different ways. First, with the stable individual traits that we can measure, for example, with the individual alpha peak frequency at rest, or with some measures that are maybe modulated more dynamically, such as the instantaneous alpha frequency, which seems to be modulated by change in alpha power. With that, I would like to thank you for your attention. And of course, thank you to David and Michael Herzog.
Thank you, Maëlan. There are any questions? We have a question from the audience. Yeah.
Thanks so much for this talk. One question. We observe
So your question was whether participants improve over time in the task. So we checked, for example, in the study with the visual backward masking, we checked that the last half of the trials, we still found the same effect than the first half of the trial. But we didn't check more than that in terms of the performance. And we know also, however, that the alpha power and the alpha frequency tend to change over time. And this is also something that we checked. We still found the effect of individual alpha peak frequency, because, so here I just presented the result with resting state individual alpha peak frequency, but we also replicated the same findings using the pre-stimulus individual alpha peak frequency. And doing this analysis, we also checked that there were no confounding effects with change in alpha power during the session. Mm-hmm. Yeah. Yeah, this is possible, but we didn't try to train individual, but it's a good idea. Yeah.
Are there any other questions? Maybe, yeah.
So, about the impact of alpha on stimulus processing, I'm guessing in your mind this extends beyond visual processing, water, stimulus, movement. I was wondering, what you think about conscious processing in states, in alternate states where you don't have alpha power, it's only in presence in the do you think it shifts by the frequencies that we normally like delta or theta? I don't know.
Yeah, I would say that if this oscillation really reflects some highly excitable and very high inhibition, we should find similar effect, but maybe with slightly different frequency. I would check first the most dominant frequency during the task or for the particular sensory processing here.
Any other questions? Okay, I have a question. Okay, is this alpha peaks also changing with age?
Yeah.
And if yes, yeah or no?
Yeah.
Okay, and in your opinion, what is the implication of the changing with the age of this? Would be the implication of this change with the age of the alpha peaks, individual alpha peaks?
So first, I know that it changed. Usually, we observe that the individual alpha peak frequency will the speed of the frequency of the alpha will increase during the first years of life, and usually it's stabilized around 20 years. And then we know also that alpha peak frequency will decrease. The speed will decrease later after 50, after 60 years old. And for example, this result that we show is individual in healthy control and schizophrenic patient. We also replicated the same findings with young and old people. Again, we also with data that we had in Michael's lab. It is the same. Older people have slower alpha peak frequency, but still we found the effect in the visual backward masking in both groups, regardless the age.
So Mattia will present his result that is about working memory, so information that is no longer in our environment. So we change a bit the topic. So Mattia is a postdoctoral scholar in the Helen Wills Neuroscience Institute at the University of California in Berkeley. Mattia works with Bob Knight, investigating the neural dynamics underlying cognitive control in the human brain. His long-term research goal is to uncover the neural architecture of distributed cognitive networks that allow us to flexibly achieve different types of cognition and behavior. Before joining the Knight Lab, Mattia completed his postdoctoral training with Mark Desposito at the University of California in Berkeley, with the support of the Swiss National Science Foundation. Before his time in Berkeley, Mattia obtained a bachelor degree in clinical engineering and a master's degree in biomedical engineering from Sapienza University in Rome, in Italy, and a PhD in psychology from the University of Fribourg here in Switzerland, completing the doctoral training of the Lemanical Neuroscience Doctoral School.
So first of all, thank you very much for having me. I'm sorry I missed the previous presentations. I'm sure they were great, and I'm gonna check them out on YouTube when you guys upload them. So my name is Mattia Pagnotta. I'm a postdoc at UC Berkeley, and I'm currently working both with Mark Desposito and Bob Knight. And as Maria mentioned, I'm broadly interested in cognitive control. So what is cognitive control? It's this fundamental group of abilities that allow us to translate our internal thoughts and goals into concrete actions. And this really consists of a triad of processes, which includes attention, our ability to allocate our limited cognitive resources at the focus of our goal, working memory, which is our fundamental ability to hold and manipulate information in mind when it's no longer present in our environment and surroundings, and goal management. And while I'm interested in the involvement of brain rhythms and oscillations in all of these processes, today I will focus particularly on working memory. So let me start with a quote about working memory. This is from my boss, Mark Desposito, and I really like this quote. I know that Matt Walker said that sleep is our superpower. While we are awake, working memory is our superpower, because it allows us to translate our knowledge into action by holding this information in mind. In this talk, I'm going to try to convince you that this incredible superpower that is working memory is supported by alpha oscillations. These dominant brain rhythms at frequencies in the range between 8-12 hertz that have been observed for over 100 years now. And I will try to make the argument that these oscillations are involved in two distinct and somewhat contradicting roles and mechanisms. On the one side, on the left side, alpha is involved in a gating mechanism that allows us to suppress irrelevant information. On the other side, this oscillation may also help us to maintain and hold information that is actually relevant during working memory maintenance. And so let me start with some background on the first mechanism. Previous studies using a task in which the cue is presented before or after the items, here represented on the left side, show that when the cue is presented before the items, here in a pre-cue condition in which basically participants are queued, are ready to attend a specific location, in this case the one where the red item is going to show up. They basically show that in these pre-cue conditions, there is an alpha lateralization effect in posterior sensors. This lateralization here is obtained by is shown by doing Q left minus Q right. And what happens is basically the studies reported a typical decrease of alpha contralateral to the hemisphere that has to be attended, hemifield that has to be attended, and increase in the ipsilateral hemifield. Some studies reported only the decrease, some studies only the increase, some the combination of both. But from these sort of experiments, all of the studies reported this strong alpha lateralization effect over posterior sides. And what is interesting is that the same pattern has been observed also after retro-Q condition. So when the Q is actually presented after the stimuli, when we are holding this information in mind and we are asking the participant basically to allocate our memory to a specific item. But these dynamics are basically accompanied also by are quicker and are also accompanied by an activation of a frontoparietal network. So there are both frontal and parietal areas involved in this mechanism, and alpha seems to play a role in suppressing information that is irrelevant by increasing, basically. My colleague Justin Riddle, who is now at Florida State University as assistant professor, when he was at Berkeley, provided causal evidence for these findings. So Justin used TMS in a retro-Q experiment very similar to the one that I showed you before. This is just the retro-Q condition. Participants were shown stimuli. After a delay, there was a retro-Q indicating whether they had to basically respond on the stimuli on the left or on the right. And then there was a probe. This is a match to sample task. And what Justin showed is that by stimulating left prefrontal cortex, help participants remembering stimuli on the right visual hemifield. This when the stimulation was in the theta band, while when the stimulation was on the left hemisphere, but in the alpha band, this helped forgetting right visual hemifield items. Again, providing causal evidence for this idea that alpha, posterior alpha may be involved in the suppression of irrelevant information, even in working memory. And he did this experiment by stimulating either frontal or parietal sides during the distinct sessions online during the execution of each trial. Stimulation was either at theta and alpha frequencies. These are the details of the stimulation patterns, or just use the stimulation condition that is basically lacks rhythmicity, that is used as a control condition. So every time you have you test the effect of rhythmicity by comparing the rhythmic stimulation, let's say in theta, to the effect on behavior in the condition that lack rhythmicity. And this is done to control for differences in stimulation sites and also across participant. Another thing that we should highlight here is that Justin stimulated right after the onset of the retro-Q. So really tapping in this quick retro-Q dynamics of alpha lateralization. Again, just as a reminder, providing supporting evidence for the idea that alpha is involved in the gating and suppression of irrelevant information. Second mechanism in which alpha oscillations are involved is actually relating to supporting working memory maintenance. And there has been supporting evidence for this from the late 90s, early 2000s. For example, studies by Wolfgang Klimesch and colleagues reported this paradoxical alpha synchronization in memory tasks. So they observed this alpha oscillations increases tracking memory information. So there seems to be some sort of contradiction between these two accounts. And we try to solve this and try to address more this sort of dichotomy of alpha oscillation. In a recent study, we use a task in which we show participant 3 circles, in this case, green, pink, and orange. And after a delay, we ask the participant to report the position of the items cued by color. So in this case, I show the 3 items. I ask the participant to report the position of the orange item, and they reported the position correctly. Something interesting happens in this kind of tasks. When I show the participant the same set of items and I ask them to report the orange, and instead they respond with the position of the green. So there is something special about these incorrect trials. These are called swaps, and these are inaccurate responses relative to target item, but are actually accurate relative to a non-target item. So these are not guesses or like random responses. There is something precise about these swap errors. And in particular, they reflect a failure to correctly binding visual features in working memory. So the correct binding of these conjunctions between color and spatial positions of these circles. A recent computational model has proposed a biological plausible way in which this feature binding is accomplished in the brain, and they suggested that this is done by synchronization of population of neurons that are selective for the different visual features. This was work by Joao Barbosa and a group of Albert Comte in Barcelona. The model is based on two ring attractor networks, one for each feature space. I'm not going to go too much into the details of the model. This is a spiking network model, so it's inspired by biology. Feature binding in this model is accomplished through the selective synchronization of pair of bumps across the two networks. So the population of neurons that represent a specific color shade like orange, and the population of neurons that represent the specific spatial location on this wheel. And this computational model is a so-called hybrid model because it combines two models of neuronal firing communication. More specifically, the maintenance of color location association is accomplished through correlated oscillatory activity, or in other words, relies on a temporal code. So temporal coding refers to a model of neuronal firing communication in which the stimulus information is encoded in the temporal structure of spike trains in terms of the relevant timing of spikes in a population of neuron, or with respect to an ongoing oscillation. So this is linked to synchronization effects in the brain, and the specific coding scheme, temporal coding, sometimes is also referred to as phase coding. However, the limitation of this temporal coding is that we require encoder and decoder for encoding and decoding information in working memory, which are able to distinguish which features are associated by detecting these ensembles oscillating in precise synchrony. So the precise timing mechanism is not quite biologically plausible. That's why this is an hybrid model, because Barbosa and colleagues, what they did is to overcome this long-standing limitation of the binding by synchrony framework. They basically propose an encoding and decoding of color location association accomplished through rate coding. This is another neural communication scheme. In the model, this is accomplished by delivering brief excitatory pulses simultaneously to each corresponding bump during encoding. So for example, the orange and the specific position, and during decoding, you just need to reactivate the population that represents orange to basically obtain the spatial position. And rate coding refers to a coding scheme in which the stimulus information is contained in the firing rate of the neurons rather than this temporal information, which means that the frequency of action potential increases as the intensity of the stimulus increases. This computational model provides a plausible neural architecture of the cortical circuits that implement feature binding in working memory. And in this model, again, temporal coding, in particular during maintenance, emerges from lateral inhibition between the different neuronal populations that store information about the distinct competing conjunctions. So let's say the specific orange spatial position against the specific green and another spatial position. Since there is in the network this lateral inhibition between the different representation, there are these emerging phase coding dynamics that are at mid frequencies in the alpha and beta band. And the central mechanism of swaps in the model is that there are abrupt changes in the phase relationship between oscillating bumps at the frequency of resonance of this network. So there are these abrupt changes due to noise in phase relationship, and this creates a jump basically on the dynamics and a misbinding between feature that ultimately at decoding results in swaps. What I liked about this model is that besides confirming previous observation from behavioral studies, like the fact that swap errors increase with delay and item competition, these are known effects in the literature. The model also provided some neurophysiological predictions. The central mechanism of swaps in the model, again, is the fact that there are these abrupt changes in the phase relationship between these oscillating bumps of population of neurons that represent the different conjunction. And the main prediction of this model is that swap trials are characterized by less phase preservation during the delay interval compared to trials characterized by high precision, in which the participants basically respond with on target. All of this was computational model and simulations. We wanted to test this prediction using real data, so we used MEG data. This was done in collaboration not only with Joao and Albert, but also with the group of Kartik Srinivasan at NYU Abu Dhabi. And what we found is that indeed the swap trials are characterized by less phase preservation during the delay. This difference here on the topography was observed over posterior electrodes contralateral to the memorized stimuli. Here I'm showing the higher phase preservation for high precision targets compared to swaps in red, contralateral to the visual stimuli. And we found that this effect was specific for alpha oscillation. So this effect was observed only at frequencies between 9, 10, and 12 Hertz. Indeed, in alpha, we observe these phase irregularities here still represented at single trial level for a swap trial. So as you can see, it's like if the alpha oscillation is almost skipping a bit, it's like the phase becomes irregular all of a sudden, and this may actually induce swaps. We also showed that swap trials are indeed characterized by higher variability in phase coding in the alpha band. This was done by using a different measure, so providing converging evidence that indeed phase coding variability may actually induce swap trials, and so as a proxy, phase coding may actually be important to maintain information and the correct binding of features in working memory. This again localized in contralateral sites. We did some reconstruction, source reconstruction also. As you can see, it's a frontoparietal network across visual areas in the hemisphere contralateral to visual stimuli. And you can check out the preprint. We also went back to the model and showed that indeed using our measures, we were able to detect these swap trials and abrupt changes in phase relationships. So in light of these discoveries, we asked, can we provide causal evidence for the role of alpha oscillations during working memory maintenance? Remember, my colleague Justin, he showed and provided causal evidence using TMS for the first mechanism of the suppression, the gating effect of alpha. Can we actually provide causal evidence for the role of alpha oscillation in helping to maintain information in memory? So let me give, before I go into the study, a big shout out to the people that made this TMS project possible. In particular, Jason Saimeka, who was a postdoc at Berkeley when I moved here 3 years ago. He really helped to set up the design and the room for running the experiment, as well as a long list of my RAs that have been helping me in the last year or so to recruit participants and collect data too. So we used for this TMS study a task similar to the one that I showed you before. However, here we asked the participant to provide 6 responses instead of only 1 report. And this was done using a color wheel. So we show 3 stimuli again, green, pink, and orange. After delay, we asked them to report the color in this case. So we queue location and we asked to report the color of a specific circle. So in this case, I'm asking the participants to report the color of this third item here, the orange one. And so we use this color wheel with the 6 responses because imagine that I ask you to report the color of the circle in the position that I just showed you, which was orange, and you have 6 chances using the color wheel. If you remember the shade of the orange with great confidence, you can basically start providing all of the responses around the same shade. And as you can see, this is an example from one trial. Participant was pretty confident about the positions. These are 6 responses stuck together. The participants were able to see the distribution. However, the color shade is quite off. It's a bit brighter than the original one. So this is important because we can derive a measure of high precision in this trial. However, the accuracy is a bit off. Other way around, imagine that I ask you to report the color shade of the pink circle here by queuing the position. In this case, actually, the accuracy was pretty good across the distribution. However, as you can see, the participant decided to spread the responses over. And they were getting points based on how close each response was to the true color shade. And we did this design because from the distribution of responses of the participants, we can fit models to derive behavioral measures of interest. So we can fit a mixture model of the participant responses to these color wheel adjuster responses. This was done using the toolbox by Paul Bayes, and we were able to derive measures in terms of number of swap trials, number of guess trials, the accuracy of the responses, as well as the precision. The precision was estimated using this measure, which is the von Mises kappa. So here, this representation, and it's the first response I showed you, it's a measure that has high precision and so high von Mises kappa. So you can imagine this as an inverse of a standard deviation of a distribution if you want. And I'm highlighting this measure because I'm gonna, just to provide a spoiler on our results, what we observe from the DMS study is that there is actually a frequency-specific modulation of the working memory precision as measured by the von Mises kappa, but not of the other measures. So what did we do? We used a design similar to the one that Justin adopted in the previous study, but here we simulated on each trial during working memory maintenance. In particular, we stimulated during the second half of memory delay with the onset of the simulation that was jittered over time for each trial. We stimulated either in middle frontal gyrus or intraparietal sulcus, so in frontal and parietal sides, always in the left hemisphere. And this was done in each participant in separate sessions, and the order of the sessions was counterbalanced across participants to try to reduce any effects of learning across the population. Stimulation intensity was set to 110% of motor thresholds for the aficionados of neurostimulation and TMS. And this was motor threshold of each participant was obtained in a preliminary session in which we were stimulating the hand knob of M1 in motor cortex, trying to get like a finger twitching. This is a pretty standard procedure. And as just in experiment, we used a stimulation in theta frequency, 1 in alpha, and 1 as a control condition that lacked rhythmicity. These were completely randomized within each block, but the same number of trials we stimulated in theta, the same number of trials we stimulated in alpha and in the arrhythmic condition. The targets were selected in MFG, middle frontal gyrus, and IPS, intraparietal sulcus, from a meta-analysis of working memory studies. And then this was done with NeuroSynth, and then the coordinates obtained from this meta-analysis of the targets were transformed onto the participants' anatomical scan. This was obtained in the preliminary session using T1 scans. So we scanned the participants with MRI, we got their anatomy. And this is important because we use also their own anatomical for line neural navigation and precise targeting during the experimental sessions, because we want to be sure that we are actually stimulating these areas in each participant. So let me go to the results. What we observe from this study, and this is still ongoing, but what we observe is that precision is increased by alpha stimulation in TMS and theta stimulation in MFG. So here in this plot, precision is shown as the von Mises kappa. Positive values are an improvement in precision compared to the control condition. Negative is basically performance is getting worse. You have on the left side the results for the premotor cortex side in MFG. On the right side, you have the results for the parietal side in IPS. Alpha is represented in this light blue, theta is represented in orange. And as you can see, we found that there is an interaction between the size and frequency of stimulation. This was found when the visual stimuli are in the visual hemifield contralateral to stimulation. However, this was not true when the stimuli are in the visual hemifield ipsilateral to stimulation. So this interaction effect disappears. So these results provide causal evidence for the involvement of posterior self oscillations contralateral to the relevant items in memory during maintenance time. So alpha helping to hold relevant information during working memory, as well as the involvement of frontal theta during maintenance. This was didn't really show the dichotomy compared to previous studies. This is in line with previous studies. So frontal theta helps contralateral to stimulation to maintain information. So together, the stimulation studies provide causal causal and converging evidence that alpha oscillations are involved in two distinct mechanisms supporting working memory function. On the one side, a gating mechanism that helps suppressing irrelevant information in working memory, but they're also involved in a second mechanism that help maintaining relevant information in working memory. On the other hand, frontal theta oscillations in working memory may help to remember and maintain relevant items regardless of the dynamics, basically, and the timing of the stimulation. So these results very exciting, but they open really many questions for us. So right now I'm asking myself, what is the simulation doing on this frontoparietal network? And to answer this question, I just joined Bob Knight's group. These are kind of my two superheroes of this story. With Bob Knight, we are started collecting intracranial data from patients that undergo surgery for medically refractory epilepsy. So we are recording directly from the brain in patients that are sitting in the hospital waiting to get surgery, and they're monitored for epilepsy. And we are implementing the same task in these patients because we want to understand exactly what the stimulation is doing in this frontoparietal network. And now it comes that accuracy is not really modulated, but for example, we have these modulations on precisions. So this is also done in collaboration with Nicole Bentley, who is a neurosurgeon at University of Alabama in Birmingham, and many other sites in the US. So this was just a second to last slide to say thank you to all of the collaborators and people that help in the projects that I show you throughout these slides. Let me just conclude by saying thank you to Maria and David for organizing this symposium. I was able actually to find this pretty old picture of David. This is from the summer of 2017 when we were both in Fribourg and we started collecting data together when David was a postdoc and I was a PhD student in Fribourg. So thank you.
So David has one.
Ciao, Mattia.
Yes.
So,
Ciao, David.
ciao. I have two questions. One is whether you think that these two mechanisms for alpha are supported actually by different types of alpha, which may also have different frequency or slightly different frequencies. And the second, which is a bit related, if I get it correctly, you usually have the approach of targeting an arbitrary rhythm as the alpha, representative 10 Hz, right? What do you expect?
Yes.
In case you could target some more, like the individual alpha peak frequency, something more specific to each individual?
Great questions. Let me just share. I feel like it may be helpful for the responses. So in terms of, let me answer first the second question. I think we are gonna see, actually, let me go back to results. These are the individual subjects. At the moment we have 14, the goal is to get to 24, 25. What I would expect is that by targeting the individual alpha frequency of each participant, we will get even stronger effects. The same for theta oscillations. Bob Knight actually has, he started a startup. It's more related to sleep studies, but they show this effect. Basically, they try to target the peak frequency of the participants. So I would expect an even stronger effect on behavior and less variability across participants, because here, some of the participants, they didn't show big differences compared to the control condition with the stimulation. So for some participants, the effect is pretty strong. For others, it's a bit, it's basically a mild effect. In terms, so I agree with your intuition of it would actually, if we were able to do preliminary non-invasive VEG, quick in non-invasive VEG, record the peak of alpha, let's say, and then stimulate at that specific frequency, I would expect the effects to be even stronger. We are planning to do that in patients, because in patients, they gave us the okay to do some stimulation. I mean, it's like the neurosurgeons are gonna do that, and they may actually be able to target specific frequencies in patients. And the idea is that then, if that works in 1 or 2 patients, we're gonna do it also with control participants, so normal, healthy subjects. In terms of the two roles of alpha, so the way I see it, rather than alpha having a role, is the fact that this is really inspired also by the model. Alpha oscillations emerge because there is some level of competition and lateral inhibition between conjunctional features or if it's in perception, visual perception of different items and whatnot. So what you have is that, I didn't show these results, but in terms of power, the hemisphere that is ipsilateral to the visual stimuli, so where you don't have information that is relevant, there is an alpha power increase that it's even higher. However, you have this alpha dynamics of temporal coding that emerge in the hemisphere that is contralateral. So the way I see it is you have locally an effect that is basically due to the fact that you're holding relevant information, and this creates lateral inhibition. Neurons are limited, so what they do is they integrate and fire, and then they have a refractory time. So the optimal way of maintaining information for population of neurons that represent a specific color shade, a specific location, is if they fire together in this temporal code. So the idea is that by having this lateral inhibition, they take turns in representing orange up, green down, orange up, green down over time. So it's an optimal way for the brain to basically use time and the resources that are available. The effect on the other hemisphere, I don't know exactly what's the source, if it's a secondary mechanism of inhibition that it's activated immediately by vision, top-down effects when you have a retro-Q information or a pre-Q information in which you know already that one has to be dropped, so you have a broad inhibition there. So there may be different sources. There may be one that is linked to the relevant items that then creates a secondary mechanism of inhibition. Ole Jensen outlined some of these hypotheses too, especially in the context of visual attention, but I feel like very similar mechanisms apply for working memory. So there may be different sources and different alpha. There is an overlapping, another thing of these different alpha sources, there may be an overlapping between the attention, so they may use the same network. However, there may be some areas, some one process and not the other.
Okay.
And the other is more on the maintenance side. These are sessions, but it's more at the moment.
Thank you, Mattia. I think the connection was just a bit off at the very end, but I think we got the response.
Okay, so thank you, Mattia.
Thank you very much, guys.
Martin Seeber obtained his PhD from Graz University of Technology, studying neural correlates of self-movement, especially gait. Afterwards, he joined the Basic Neuroscience Department at Campus Biotech in Geneva, in Switzerland, to investigate intrinsic spontaneous brain dynamics by combining intracranial and scalp electroencephalography. At UCLA, so University of California in Los Angeles, he aims to better understand the neural dynamics related to movement trajectory and the formation of episodic knowledge with the aid of intracranial EEG and full body motion capture. So the stage is yours. Thank you, Martin.
Thank you. Also, thank you for having me, Maria and David, and thank you for organizing the webinar. It's good to be in touch, good old times. I have good memories to Geneva, and I'm happy to talk a little bit more about theta dynamics, following up Mattia's fantastic talk, where we talk about theta dynamics in the human medial temporal lobe, maybe completing then the networking, then going back to the prefrontal cortex in the very end of my talk. So I'm going to touch upon human theta dynamics during real-world and imagined navigation, which is a little bit a different topic, but maybe we can discuss some parallels between visual processing and navigation and episodic memory, maybe in the very end. Let's see if we can flip. All this work is heavily inspired by works by Bujaki and Moser, where they discussed a long-standing vision or perspective in neuroscience that actually episodic memory or memory in general might have evolved from navigational abilities. So you can imagine that the way we navigate in space is very similar on a conceptual level of how we remember episodic memories, meaning they're how we navigate between specific points here, between different islands in this example, might be very similar of how we connect past events and the succession of these events to remember a particular episode in our life. And of course, because those are neuroscientists, they do not only compare these two tasks on a conceptual level, but describe the neuronal basis of the tasks. And to describe these parallels of spatially tuned neurons in the hippocampus are essential. And as most of you already know, that there are these place cells that fire in a sequence when an animal is running on a specific path. So there are a number of neurons here sorted by their firing depending on positions. And you can see this very beautiful, like, position-dependent specificity here. And these activities, so these neural sequences, are not only present when animals are running through space, but actually are also present when animals are navigating between two different routes and come back running this wheel. And even during wheel running, there are well-organized neuronal sequences that are informative to their next choices. And that really fascinated me, because this internal generation, or the ability to internally generate sequences that is independent of environmental cues or strong sensory inputs, that might actually be then the bridge between navigation and memory, because you can imagine that during memory, we do not have strong sensory inputs, so there must be an internal mechanism to generate these sequences. And for this generation, theta oscillations are crucial, because if you because these theta oscillations organize these temporal sequences, meaning providing a temporal frame to the ordering of this sequential firing in animals. And that might be also true in humans. In animal studies, if you inhibit these theta oscillations, memory performance goes down. So these theta oscillations, they might have a causal role in the establishment of these neural sequences. But what we wanted to test is whether these theta oscillations are present and organized in human real navigation, because previous studies found that these theta oscillations are not continuous in humans, but instead they appear in intermittent bouts. So instead of having continuous oscillations providing these time frames, these oscillations might be more intermittent and more infrequent. So we wanted to test the functional significance of these theta oscillations in humans while people are actually moving around and remembering their routes. And this research is only possible by developments that were taking place in our lab, where in 2013, I think, the responsive neurostimulation device from NeuroPace was approved from the FDA. And that is initially was developed to treat epilepsy in participants who are treatment resistant, but also these stimulation device can record brain activity in order to stimulate the brain specifically after detecting epileptic events. So this device can also then be used to record brain data after developing specific research tools. And these research tools were developed only recently here in this publication, presented in this publication by a colleague of mine, that is Urst Pavlovic, and was published already 4 years ago. And what the colleagues here did is developing a mobile setup that can actually access this neurostimulation device and record brain activities from the medial temporal lobe, so deep brain regions, while people are freely behaving in space. And that gives us the ability to study natural behavior in humans while they freely move and can mobile interact with their environment. So this is just a video showing the technology. You can see here the person is moving around. You can see here the intracranial EEG recordings on the bottom right, the position in space from top down. Here you can see eye movements and the first person perspective of these participants. Because we go from a stationary experimental setup to a mobile experimental setup, we have to record as many behavioral variables we can get to rule out potential confounds. But when I saw this, I was very excited that this is actually possible, and this is the reason why I moved to LA to conduct this research. So following up on the introduction I was outlining, we designed an experiment very similar to previous animal recordings where we showed this schematic here presented on the left to participants and asked them to navigate these two specific routes. So it's a left and a rightward route. They were asked to do in this motion tracking space, and nothing in the room really indicates where these routes are. So we just show them these routes, and then they have to remember where in the room they have to go in order to walk exactly, precisely on this route. And you can see here on the right that this person was equipped with motion tracking and also this wand, we call it, in the backpack that records and streams the neural recording and also the behavioral variables. In order that we can then compare their motion trajectories and neural activities, which we record from the medial temporal lobe, and more particularly, I'm going to talk about the anterior hippocampal recordings in this experiment. And here on the right, I show raw data in gray and filtered data in the theta range in blue. And you can see here that these oscillations, they're not strong and then like get stronger in these spots. And then vanish again and come again. So we wanted to investigate this temporal structure depending on position as a first starting point, because this is still one of the first mobile experiments in humans using intracranial data. And the paper was also recently accepted for publication, so I'm very happy for that and looking forward for the publication to be out. As I said, people were learning this task. So first we just showed them this schematic. And you can see here in these light gray lines, first they didn't really nail it. They walked roughly around the room, but didn't hit the right angles and distances of these route segments. But after each round, they come back and I showed them their behavior performance after each completion of the run. And then they did better and better. And you can see here also after 5 runs, they did really quite well. So this like dark gray line is the 5th trial, which is like very close to the perfect one. And they got better and better in time. In total, I recorded about 40 gait trials, and after each actual walks, they came back on the treadmill and only walked on the treadmill without any additional task, which is the control condition in our experiment. Or after the first session and sufficient learning, I asked them then to imagine navigating the exact same routes in their mind. So what we have here is then the real-world behavior of a real-world navigation, a control condition that is like only walking, and walking while navigating in their minds. And the gist of the experiment is that when they walk on a treadmill, they either navigate or not, while the behavior is exactly the same, like their motion behavior. So let's first look at the neural activity in comparison to behavior. Here on the left, I show the motion trajectories of all 5 participants included in the study. And as you can see that these average trajectories are very, very similar to the ideal one. So they really learned to perform this task well after we provided them some feedback. And on the right-hand side, I show a time frequency plot of the left and right walks. Here is the start of the experiment and here is the end. So it took them at about 28 seconds to complete one route. And this is the average across all trials. And you already can see there is some specificity, both in time and frequency. And at around, in the theta range, at around 4 until 9 hertz, you can see these bouts coming up. And these bouts are aligned to specific instances in their task performance. And these instances, they coincide with the turns. So if you then plot the averaged theta activity in this frequency band, depending on the position, so here we combine the motion tracking and the neural data to illustrate both types of data in one image. Here in color coded, you already can see by naked eye that whenever a participant is completing a segment and approaching a turn, theta power goes up. And that is true almost in every segment and also in all of the participants. So this is a group average showing an average over all medial temporal lobe channels. And if you then average all the data depending on the turn, so if you align then all neural activity depending on the turn, when the turn is in the future at 0, and then we go back in time and look at the time frequency activities, you already can see at around -2 seconds, the theta activities, they go up already 2 seconds before they actually perform this physical turn. And then this could also be very trivial behavioral confound. So we looked at behavioral data where we show physical turning here in this curve the velocity here on the top. So whenever they approach a turn, they slow down in order to do the physical turn to go in the right direction. But even before the first slightest decrease of velocity, theta already is almost at its maximum. So all these neural signals, they precede the behavioral variables. So so that is almost like ruling out a trivial behavioral confound by these turning signals and slowing down effects. And this effect was also present in all of the participants which we included in our study. So I should already show you the group data, but for participant 1, 2, 3, 4, 5, it's a very, very similar pattern in each participant. Here I show one electrode per participant in the left anterior hippocampus. And remarkably, that participant 2, for some reason, didn't learn the pattern perfectly, despite the fact we instructed them repetitively, but did this additional turn here on this fourth root segment of the left walks. But despite the fact that this was not instructed, the neural activity still showed this approach, turn approach effect here in this like theta increase on this additional turn, somehow proving a point. Whenever they learned to complete the segment and transition between one segment to the next, there is this theta power going up. And that also can be illustrated in a video. What I'm showing here is a participant walking. On the bottom, there is a lower body skeleton fit where you can see the leg movements and the precise position here from a top-down perspective. So show the movement trajectory, and on the right, theta power just as a bar plot, and below is the raw EG of one particular channel. And as a participant is walking and approaching a turn, you can see these oscillatory bouts coming up that can be seen in the raw signal. Again, there is like another turn in the end, and you can see these recordings to get much more oscillatory, and these are really like bout-like activities. You can see here almost like a desynchronized pattern, but then these oscillations are like getting much stronger and much more oscillatory, which is also true for turns they take in the middle of the room. So it's not related to approaching a wall or a specific boundary, which I found very fascinating. So that is the relation between theta activity in the brain and real-world behavior or real-world navigation. And we also looked at the single trials. So here I'm plotting 36 single trials and then color-coded the amplitude of theta activities. So this is theta activities depending on trials. And if you stack all the trials on top of them, you already can see with naked eye again these clusters of theta bouts, we call them. And these clusters, they correspond to these turns. So to quantify the temporal consistency of this data, we split the trials in two parts, A and B, and then we average over A and over B and compare the signals to get a measure of how consistent actually this single trial temporal structure is in the data. So if there would be just like no structure, if these theta bouts would appear only stochastically, this correlation would of course be zero. But if there is any kind of temporal structure in the data, these two parts would be highly correlated. And we do not only do that for the first and the second part in this example, but randomly pick half of the trials for pool A and half of the trials for pool B to get a more robust estimate of this temporal consistency measure. And if we look across the three conditions, we see no temporal consistency in the control condition when they only walk in the treadmill but do not navigate at all. So this is data shown from 18 channels, all 18 channels included in the study. So they really nicely cluster around zero. So there's no temporal structure during sole treadmill walking. There is solid temporal structure during real world walking here. That is also significant than in the control condition. And during imagination, there is something in between. Meaning, if they walk on a treadmill, only imagine their navigations, there is a significant temporal structure in their neural data, despite the fact they do not really move through space. They are just stationary and imagine themselves moving along these routes. And that I found quite remarkable. And it also makes sense that somehow it's in between. It's not as good as in real-world navigation because there is less rigid temporal structure in their imaginations than if they would walk in the real life. And looking at the different channels, we also correlated looked at whether the strongest temporally consistent channels are the ones during in navigation are the ones that also corresponds most strongly during the imagination condition, and that is actually the case. So the channels that do not show any kind of temporal structure during real-world navigation also have like low temporal consistency during imagined navigation, and this cluster of actually these 5 left anterior hippocampal channels, this is like 1 channel per participant, they form a cluster with like the strongest temporal consistency. So that seemed to be like an anatomical structure that is like most responsible to produce this temporally structured dynamics. And then what we did is looking at the neural activities during imagined navigation too, and we were all surprised that this temporal structure was like surprisingly well aligned to the trajectory. So how we produce this plot here is only by using the time, meaning the start and the end of their imaginations. So the start, we told them to start, but the end they reported with a button press. So we know the timing of their imaginations. We do not know so much the turns in between, but what we do know is what we have like a solid estimate for is this very first turn and this very last turns where we see these activities are somewhat similar. So we wanted to quantify and investigate these similarities in more detail. To do so, we looked first at all the electrodes being aligned. So taking this neural data of the real-world navigation condition, we then aligned all the segments. Because segments have different lengths, we stretched them in a way that all the segments have the same length, and then looked at the variability across all of these 18 channels of different people. And that is what I'm plotting here. You can see here relative position would be like a segment would start here at 0, and the end of the segment will be here at 100%. So you can see here that, as I described, you can see in this plot that all the different channels, they seem to increase their power towards the end of a segment or ahead of a turn that transitions from one segment to the other. But as different channels, they have different alignment. So we thought, looking at this data, using these different alignments, some channels, they have more like a cosine alignment, and some other channels, they might align more to like a minus sinusoid pattern. So that is somehow the abstraction we took from this observation of the real-world navigation data, and we made a model combining this cosine model, we call it, or cosine root model, and the minus sine model, because a cosine and a sine, they can result in circular activities. And so the intention here is to reconstruct the relative position within one segment, not only at the end, but covering the whole segment. And that is what we did using a simple regression model. So we tried to compress all these neural data from these 18 channels in these 2 different latent dynamics, and then use this latent dynamics to estimate their position. And that also worked. I mean, it must work in the real-world navigation condition because we designed the model accordingly. But the important data point is here that it also worked for the imagined navigation data, meaning here for this sine reconstructed dynamics, these are aligned very beautifully as it should be in the model, and this is cross-validated data. So it's data that only generalizes the training. So this is data that is independent of the training of the model. So we set up this model to map neural activities to relative positions during real-world navigation, and then we apply this neural decoder to the imagined navigation data. And what we see is also this like 4-parted modulation that is very similar during imagined navigation than in the real-world condition. And then when we cross-correlate this model dynamics while correcting for the different length of the segments, we would expect a 5 times repetitive pattern, and this is also what we see. So when we correlate this model with the actual decoded signals using again cross-validation, then we see very high correspondence between the actual model and neural data, not only for the real world navigation, but also for the imagined navigation condition. So that would be like the correspondence between the model at point 0. If you shift the data by 1 segment, 2, 3, and so forth for segments. And that is because it's 4 peaks, because they're like 4 turns in these modulations. And that we did not find in the control condition. So this is like not significant correspondence or even resonance, if you will, so to this root structure we formalized here. And then using this model, then we also can reconstruct positions. And here I'm showing actual positions used like recorded from motion tracking. So again, 0 until 100% within 1 segment. And what I'm showing here is a histogram of like how likely is it that we are actually reconstructing the positions, right? And perfect would be like a diagonal. And you can see it's quite diagonal in the real world navigation conditions. So the more red, the more right we are. So in how many cases are we like better than chance, or in how many cases, given we have like, for instance, the measured position of 40% in the relative position within 1 segment, how often do we actually reconstruct 40% then just giving the neural data? So that works nicely in the real world navigation condition, but also to some extent in the imagined navigation, where it works better like around the beginnings and the ends of the segments. But again, there is a clustering around the diagonal, which is not there in the control condition that is so treadmill walking. And if you then more mathematically estimate the errors, here we have an estimation of position as like a phase metric. So this is why those are polar plots. You can see that the difference between actual and estimated positions, they cluster here around 0 for both navigational conditions, but not during the control condition. And going forward, one future aim of our research is not only to study the hippocampus here using this intracranial data, but also completing the network using scalp EEG and estimating cortical signals. So this is what we try to do next. And as most of us, Maria, me, and David did in Geneva, is to use the scalp EEG to compute cortical sources and investigate then the dynamics between different sites in the brain. And that's exactly what we try to continue here. So we also recorded scalp EEG simultaneously with this intracranial data, and we established a model we derived from magnetic resonance imaging and co-registered electrodes and identified specific structures. Here I'm showing left and right hippocampus overlaid in their anatomy. And what we found is already as preliminary results is along with these electrodes, here I'm showing the intracranial electrodes with these blue dots here, we can then look at which areas in the brain using source imaging, here Ilureta, correspond best to the ground truth intracranial signals. And as previously shown for the thalamus, we also can show that for the hippocampus now, is that we can actually reconstruct these deep sources, even only using 64 channels in mobile situations. And for people who are interested more in topographies, we always see these smooth topographies that are very distributed across the whole scalp that correspond the best with these deep sources. Here I'm showing an example of the left anterior hippocampus again, because those are the functional data, correspond to the functional data I showed before. And then now the interesting part comes about that we not only can then try to reconstruct the signals we actually recorded in the brain, but other signals that are more cortical. So we also found prefrontal cortex sources, which are much easier to construct in non-invasive data. And what I'm here showing on the right is just the completion of the story I told before. So this is a correspondence. It's just like simple cross-correlation between the intracranial EEG, so theta envelopes recorded from or gained from the intracranial data, and reconstructed scalp MTL data in the left hemisphere, and investigating whether those are significantly correlated and what the timing is. So here the timing is almost 0, which is indicating it's either volume conduction or a source that is very close to it. There could also be entorhinal cortex data or anything around, and that doesn't have to be necessarily exactly that signal. But I mean, still we have like on average across participants a correlation between non-invasively reconstructed and ground truth signals of like 0.4, which I believe is like surprisingly high. And then we can correlate or try to correlate then the intracranial signals recorded in the hippocampus and these cortical prefrontal sources we gain from scalp EEG to do some kind of like first steps towards network identification or network investigation approaches. And also between these hippocampal sources, here again, intracranial EEG correlated with the scalp EEG is significantly correlated. It's more variable across participants, but this is like a first step, not only to study the hippocampus during mobile experiments, but also to study networks that are specific to these intracranial recordings. So we somehow think of like studying the subnetworks that are spanned, they are functionally spanned from these intracranial signals and are most relevant to these nodes. And with that, I already want to summarize up. Theta oscillations in the human MTL are intermittent and temporarily structured during navigation. So that was already a surprise that despite the fact that theta oscillations are not continuous in humans, they are structured in a way that they relate to task-relevant instances. So in our case, these theta oscillations encoded root segments similarly during real-world and imagined navigation, despite the fact that there are no environmental constraints or very strong sensory input that can drive these oscillations during imagination, leading to our conclusion that this internally generated theta dynamics, they might support imagination, but also episodic memory recollection when there is minimal sensory input, which is a very important piece of evidence for bridging navigation and episodic memory or memory retrieval. And going forward, that is the combined intracranial and scalp EEG enable novel perspectives to study cortical, but also deep brain activities in motorized setups in a perspective that using then scalp EEG reconstructions, then we also can include more participants, complete networks by using the intracranial data in participants we have intracranial data, but also gives us the ability to study more clinical and larger populations and address these subcortical cortical networks in people suffering from neurological conditions. And with that, I would like to thank my supervisor, that is Nathi Uthana, my amazing lab who supported me throughout this project, all our clinical partners and funding agencies. The paper is already accepted and will be out soon. Here is my contact information. If you can't wait to read the paper, here is the preprint available. I'm happy to discuss any questions. And thank you for you, Maria and David, to organize this webinar.
Okay, thank you, Martin, for your talk. Are there any questions from the audience? Okay, in the meantime, I will have a question for you. So today we talk a lot about, let's say, individual rhythms. So one question is, also in your study that you presented, that was really fascinating, in particular for the part of the imagination, do you think that in order to achieve also better results also in your study, you can sort of estimate a certain individual range for the theta rhythm for each of your participants, for instance?
Yes, so that is exactly what I did. I mean, I just didn't have time. I look first at the spectral peaks of these theta oscillations in the medial temporal lobe, and then within the range, like within the broader range of like 3 until 9 hertz, I just select a smaller range like around this peak for each individuals. It does not, so it doesn't really drive, it doesn't make a difference if you use just 4 until 8, which is the standard bandwidth. But I think it's more precise to individualize these frequency ranges to really investigate the phenomenon that are there in each participant, as long as they're classed in the range where we expect them to have.
Good. Okay, so relating to that, do you find also some relationship between the individual theta rhythms and some specific characteristic of the patient, or they're, they were within the classical theta range, or maybe there was an association with, I don't know if their patients with their disease or their age or stuff like that?
Yes, I mean, it's an interesting question. Here we just have 5 participants, so it's we don't have enough data to make this
Okay.
conclusion. The only piece of evidence or piece of it's just like a hint, basically, what I have is that this participant who learned a little bit had a harder time to learn, had
Yeah.
slower rhythms throughout the medial temporal lobe, and also had some complaints in their learning abilities. But we did not quantify their, other cognitive abilities in addition, like apart from these navigational abilities. But I mean, still, even though the fact, that in some participants, these frequencies, they peak at 8 hertz, and in other participants, rather it's like 4 or 5 hertz, the functional, the modulation given the task is very, very similar, which is surprising to me.
Thank you, Martin. Do we have any other question? Okay, probably the audience is destroyed after
No,
one.
I know it's a
Yeah.
long day for you guys already here. I'm just
Yeah, your day is just started, ours is almost ending. So thank you. Thank you,
all the
Martin.
best. Warm greetings to everybody to Lausanne, right?
Yeah.
Thank you, Martin.